Hello everyone. I'm very glad to participate to this great conference. So I'm going to present preliminary results on the variability of vertebral trabecular bone structure. So why are we so interested in the vertebral column? First, because it's a very interesting region for reconstructing locomotor behaviors. Indeed, the vertebral column has a pivotal role in posture, overall trunk stability and mobility, but also locomotion. Comparative studies highlighted that the long, flexible, and lordotic spine of extant humans was quite compatible with our obligatory terrestrial bipedalism, while the short, relatively immobile and straight column of other great apes was more compatible with their arboreal activities. One region of the vertebrae, the body, is particularly of interest because it's considered as the main weight-bearing portion of the vertebra. We know that the inner bone organization at specific sites locally reflects the nature, the direction, the frequency, and magnitude of stresses, such as biomechanical loads related to locomotion. So, what about in the vertebrae? The trabecular network within the body has actually been demonstrated to discriminate, to some extent, different locomotor behaviors in great apes. However, the nature and magnitude of inner structural variation within a vertebra or along the spine is still debated in the literature. Evolutionary speaking, the vertebral column is quite interesting and intriguing. For instance, the timing and mode of emergence of the extant human spine still remains unknown. And in the hominin fossil record, mostly isolated or fragmented vertebrae were found, with, however, a few specimens with significant part of the vertebral column. However, despite the discovery of these, several aspects of the early hominin spine are still highly disputed, notably because of the difficult identification of isolated, fragmented, or partial vertebra, but also because of the many debates and contentious on how to define the position of a specific vertebra in the column. For example, the modal number of thoracic and lumbar vertebrae in early hominins still remains quite debated. Lastly, another challenge is the stratigraphic association of hominin remains with large papionins, which of course complicates the taxonomical assignment of isolated vertebrae. So, our study aims to explore the trabecular bone structure in the bodies of vertebrae of extant human and non-human primates, and knowledge of this variation would help to taxonomically and anatomically identify isolated vertebrae and potentially infer early hominin locomotor behavior. Our sample includes micro-CT scans of complete vertebral columns with cervical, thoracic, and lumbar vertebrae for each individual. We had 3 primate taxa having various locomotor behaviors: 4 Homo sapiens, 4 pan or chimpanzees, 4 papio baboons. Following micro-CT scanning, the first step was to define and extract 3D volumes of interest from each vertebrae. And once extracted, these voids were carefully segmented. From this segmented void, trabecular bone structural properties were recorded. First, the bone volume fraction, approximating the trabecular bone density, then the trabecular thickness, the trabecular spacing, measuring the average trabecular separation, and finally the degree of anisotropy, evaluating the average orientation of the trabecular struts. So, onto our results now. First, we'll try to answer to the following question: how does the trabecular bone vary along the spine itself and within each species? Our preliminary results in humans highlighted significant differences between segments, with greater trabecular bone density and lower trabecular spacing in the cervical vertebrae compared to thoracics and lumbars. And the degree of anisotropy was also smaller in cervical versus lumbar. In chimpanzees, similar results were observed for BVTV and TBSP in the cervicals, while for the orientation, thoracic vertebrae this time showed a greater degree of anisotropy. Finally, in baboons, We had, for the lumbars, significantly lower density values, with trabeculae more widely spaced compared to the other segments. And on the other hand, trabeculae were the thickest in cervical vertebrae. Finally, interestingly, the degree of anisotropy was significantly different between all three segments and increasing towards the lumbars. Let's move on to the second part of our results. How does the trabecular bone vary between species? So first, in the cervical vertebrae, we can notice thicker trabeculae in Homo compared to Pan and Papio, but also a more widely spaced network. Regarding the degree of anisotropy, DA is much higher in chimpanzees compared to Homo and Papio. In the thoracic segment, differences between species are quite marked, while the pattern stays similar, with the thickest and well-spaced trabeculae in Homo compared to Pan and Papio. The degree of, as in anisotropy, is again greater in Pan than in Papio and humans. Lastly, though, we can observe a difference in the trabecular densities, with a greater density in baboons compared to the two others. Lastly, for the lumbars, we have again the pattern seen in the cervicals, with thicker and more spaced trabeculae in humans compared to chimpanzees and baboons. And for the degree of anisotropy, Papio seems to now have the greater values. So if we sum up our results and try to answer to our two questions, firstly, we can say that in humans and chimpanzees, the differences are particularly pronounced between the cervical vertebrae, with the densest, well-packed and less organized trabecular network than in the rest of the spine. This could actually indicate adaptation of the cervical microstructure to the specific role of the cervical vertebrae within the spine and within the skeleton, like providing greater mobility and range of movement. In Papio, however, the parameters in the lumbars seems to stand out a bit more, with the densest, well-spaced and better organized trabecular network compared to humans and chimpanzees. This could actually reflect the specificities of the lumbar region in long-backed primates like Papio to the shorter and stiffer lumbar segments of hominoids. Our second question was about interspecific variability. First thing, the bone volume fraction or bone density was quite similar across all three species in both cervical and lumbar regions. The trabeculae themselves were quite different, particularly in humans. We can see that along the entire spine, trabeculae are thicker, more spaced, and less organized in Homo compared to the two other species, and particularly compared to Papio, which is almost always the complete opposite. So this exploratory and quantitative approach highlights the potential of vertebral trabecular bone to anatomically and taxonomically identify isolated or fragmentary vertebrae. And furthermore, the observed differences between species could reflect distinct biomechanical loads, and the identification on functional signals able to discriminate them would really contribute to discussion on the reconstruction of the locomotor behavior of early hominids. This study is still preliminary and part of an ongoing project. We are currently busy testing the methods themselves, exploring a more global method that could provide structural information on the whole vertebral body and not just in voice, like you can see in the map here. We are also working on increasing the sample with more individuals, more hominoid species with gorillas, pongo, ilobates, but also papionins to encompass more various locomotor behaviors. And lastly, our end goals is to apply this methodology to fossil specimens. So if you are interested, we will present our results on STW841, an Australopithecus africanus thoracolumbar series from Sterkfontein in South Africa, at the AABA next week. Thank you for listening. Thanks to everyone who made this conference possible, and particular thanks to Amélie Baudet for this awesome research project.